Okay, welcome to a lecture video for Chapter 5. Typically, I haven't had one of these, but it was requested, so here you go. You know I skipped through some of this stuff right off the bat. We are going for the membrane. So pretty much here, the main part to understand is the fluid mosaic model. In class, I tend to ask you if you know what a mosaic is. And mosaic is basically a type of art where you put a lot of little pieces together in order to make the whole. The example I use is Cinderella's castle at Disney World. When you walk through it, they have this gorgeous, gorgeous mosaic all over the walls. And so that's pretty much what's going on here for the membrane structure. You have all these little pieces that go together in order to make the whole. Fluid because, movable. So this is what happens for the phospholipid bilayer. This is where you will have proteins just attached in there in order to help things go in and out of the cell. The plasma membrane is known as selective permeability. What that means is it's deciding what can go through and what cannot. So then here are a variety of the proteins. Not going to go into detail on them. Picture pretty much tells you, and it goes more into it in just a moment. Main thing to notice here, all of these are different types of proteins. They all have different names by how they work. The main ones I'm going to point out are, let's do the laser pointer. So right here, we've got the enzymes because enzymes are proteins. So what happens with the enzymes is it makes something speed up. It makes a reaction happen. So if you notice here, it has one coming in, it changes it to this, which then triggers that, which gives you the final thing. We've got more pictures in a moment. Enzymes also have what I call the lock and key method. So it has to be a very specific something that is that exact shape to make it happen. And then down here, I'm going to point to the transport proteins. You have the ones where it just goes right on through channel. It doesn't really take any effort. It just goes right through. Active. Active means it requires energy, which is why you see right here ATP, because we have to remember ATP means energy. Diffusion of the small nonpolar molecules, it just happens. So the gases just go in and out to their little heart's content. They don't need help. And this is breaking it down. Oh, let me move that out of the way. This is breaking down each of the ones we just looked at. So take a moment to take a look, but I don't go over every one of these. And remember, this PowerPoint is available in your class, so you can just download it whenever you wish. Okay, so the phospholipids are going to self-assemble into the simple membrane, mainly because what we've talked about before, we've got the hydrophilic heads and the hydrophobic tails. So they automatically connect this way because what loves water is on the outside, what hates water is on the inside. And it's not that it loves or hates, but that's literally what the words mean. So hydrophilic, hydro for water, philic, love. That's why they're on the outside. Hydrophobic, water, hydrophobic, fear. Those are on the middle. So that's how it makes those. Diffusion. This is where it's going to spread out evenly in an available space. These gas particles or whatever. Happens all on its own, does not require energy. So this is known as passive transport. It's going to go from a area of high concentration into an area of low concentration. And that's how it works. And I used a very specific example in class. And we talked about it again in lab. This is just trying to show you what happens where the molecules, where there's a whole bunch on one side, move on over to the other because they're trying to equalize. So the entire goal of diffusion is equilibrium. It wants the same amount of molecules on both sides. And this is just showing you it happens both directions. Now, diffusion is just the whole gas part. Osmosis is diffusion of water across the selectively permeable membrane. So water is what's moving, not the gas, just water. So that's what osmosis is. And so that's what brings us to all of this. So now we have this little image and it's showing you that we have a whole bunch of molecules over here, solutes. And on this side, well, I guess I should do this. This has a high concentration of solutes. This has a low concentration of solutes. But the solutes cannot go through the membrane. The only thing that can go through the membrane is water. So in order to equalize, the water moves over here to try to make it as even as possible. This is an example of osmosis. And for us in the lab, we did it with our eggs. Tonicity. Tonicity is a term that describes the ability of a surrounding solution to cause a cell to gain or lose water, just like with the eggs. This is how we were talking about hypertonic, hypotonic, and isotonic. Hyper, high, tonic, solute. So that means a higher amount of solutes in the solution. Hypotonic solution, hypo, low, tonic, solutes. So this means the solution has a lower number of solutes. And then iso just means same. So isotonic solution means it's the same on both sides. We spent some time in class talking about this, and I pointed out for the plant cell, you want turgid, you want it to have hypotonic solution. So lower solutes in the solution, because this is what makes the plant nice and firm. The example I used for this, I went on and on. I did a whole story about in the grocery store. You go into the produce department. If it's Kroger, they do that. And they mist all the produce. And you pick up that lettuce, you shake it off a little bit because you don't want all that water. You put it in your bag, put it in the cart, and you go about your shopping. You finish shopping, you put it in the trunk of your car. It's kind of warm. You get home, you notice that inside the plastic bag, there's a bunch of water. You just shove it in the fridge. And then the next day you pull out your lettuce and it's like, it's not as cool as it seemed. That's because in the grocery store, they're constantly misting it with the water, and that's making it turgid. That's why it looks good. It starts to lose this as it's losing the water. That's where it starts to become flaccid, and it's like bleh. So that's the example I gave you in class. For the animal cells, this is part of why the doctors are constantly, and only part of why the doctors are constantly drilling into people, lower your sodium intake, increase your water. Because the normal, I'm trying to get this here, this one, the normal red blood cell is what we need. We need there to be the same amount of sodium inside as well as outside. So then these red blood cells work normally, because these guys are what's bringing the oxygen and nutrients through our entire body and removing the waste and the carbon dioxide out. If our blood cells aren't working properly, if they're not healthy, it impedes what they can do. Hypotonic means you don't have enough sodium in your body. And at this point, the cells are trying to equalize. And so they're pulling water from your body into the blood cell, and it can cause them to lyse, go and just pop. Not good. This is the one I have a tendency to do. I drink a lot of water. I don't do a whole lot of sodium. So I have to keep an eye on this. And let me go ahead and just say, it's not that you can drink too much water. It's you also need to eat food. You need to have a healthy diet. You're not going to hurt yourself by drinking too much water. You can very much damage yourself by not drinking enough. Which brings us here to the hypertonic solution. This one is when people are not drinking enough water. They have too much sodium. And so the body starts pulling the water out of your blood cells, trying to equalize. This is where you start to get more bloating. You get the cankles, your rings don't fit. You take off your socks and you have a line around your leg because it's pressed in because you're swelling. Your shoes don't fit. Things like that. That's due to the hypertonic solution in your body. You drink more water. Yes, you're going to pee, but that's not a bad thing. And you will find that that bloating disappears because you're helping your blood. It starts to equalize. Why this is dangerous, other than the fact of it's really not comfortable, is as it's pulling all the water out of your blood cells, it's shrinking the blood cells. They can't function the way they're supposed to, and they kind of can start traffic jams in your veins and arteries, which causes a lot of health risk. So proper hydration is key. Okay, we have talked about the hydrophobic stuff. The hydrophobic substances easily diffuse across a cell membrane because they're trying to get out. Polar or charged substances do not easily cross, so they need help. Yeah. So here we go. We've got facilitated diffusion. Facilitated diffusion needs help, but does not need energy. So you have to facilitate the movement, and it tends to be the concentration gradient. So that would be high concentration to low concentration. I skipped over this in class too, so don't worry. So just showing you the movement. The Rh protein we get into more when we hit genetics and we talk about blood. So don't worry too much about that. Active transport. Active means it needs energy because in order to be active, you require energy. That's the main thing you have to remember. So if a cell, if something's going against the concentration gradient, the cell has to use energy to make it happen. And this is an example of going across the gradient. So you will notice right here, we've got our solute that goes into the transport protein. Energy is required to slap this sucker closed and shove it out the other side. And then it goes back to the original shape and waits for more. So that is an example of an active diffusion. That brings us to exocytosis and endocytosis. EX, exit. EN, enter. Cyte means cell. So when you see EX in cyte, that means outside the cell. So it is putting it outside of the cell. EN for enter, CYT for cell, that means it's going into the cell. That's the main way to remember it. Now, endocytosis, bringing stuff into the cell. We have phagocytosis, where it engulfs, and we have receptor-mediated endocytosis. And we're going to have some pictures. So here we go. The top one, this one right here, this is phagocytosis. It just throws out these little pseudopods. Pseudo means false. Pod is going to be for foot. So it's these little fake feet that come out and then pull it in with what it needs. So that is phagocytosis, pulls it in. Receptor mediated, it has these little receptors that they're only going to react to very specific molecules. So when those molecules attach, then it closes up and pulls it in. Okay, so now we get to the energy portion. Kinetic energy, energy of motion. Potential energy is energy that's stored and it has not been used yet, but potential energy turns into kinetic energy. Chemical energy is a type of potential energy, but not all potential energy is chemical energy. I hope that makes sense. Potential energy, chemical energy turn into kinetic energy. Then we have our first and second laws of thermodynamics. The first law of thermodynamics is that energy can change form, but cannot be created or destroyed. So this means that energy is not created or destroyed ever. It just gets transferred. That's the first law. Second law of thermodynamics is as it transfers, it's losing stuff, and that energy is lost as heat. When it's losing the energy, it's increasing disorder, which is called entropy. In class, what I tend to do is I then say to put your hands together, because when it's like this is potential energy. As you go like this is kinetic energy. And what you feel is in between your hands, they start to warm up. So that is the loss of heat. So this is an example of the laws of thermodynamics. I'm not creating the energy, I'm not losing the energy, but it is transferring and it is having a byproduct of heat. So that's what I do. Okay, so 2 laws of thermodynamics. We have these examples here where it's comparing a car to a cell. For the car, it takes in gasoline and oxygen. It converts everything throughout the energy, and what comes out is carbon dioxide and water. Very similar to how our cell works, except our cell takes in glucose, which is sugar. So our cell takes in the sugar and oxygen. It then goes through the cell, converts it to what we need, and it comes out in carbon dioxide and water. Here are those words again. EX for exit, EN for enter. So exergonic reaction is releasing energy. Endergonic reaction requires energy. And that's pretty much it. Then we have a bunch of graphs that I don't worry about. ATP is where I remind you ATP means energy. ATP powers almost all the forms of cellular work, so you have to have ATP. And then this is just to show you ATP, the TP means triphosphate. You can have the water come in here and break it, which if you remember is hydrolysis, or hydrolysis is the proper way to say it. I say hydrolysis so I can remember water cuts. And this gives you a DP. The D stands for di, which means 2. And then this is just showing you different ways that this works. Now, enzymes, I already told you enzymes are protein catalysts. And so this causes things to happen faster. And that's all this is. So it's going to require a little bit less as long as you have the enzyme. This part is where I spoke previously about enzymes having a lock and key mechanism. So that's all this is showing, that the active side of the enzyme requires a very specific something. That substrate fits perfectly. The water comes in, breaks the bond, and then it releases the products. So that's all that's showing. This is a good thing to understand just because it's kind of interesting. So we have a competitive inhibitor and a non-competitive inhibitor. Competitive inhibitor means that it is competing with that very specific substrate. The non-competitive inhibitor leaves that area alone, but it mucks up the shape of everything. So now the enzyme can't pick up what it requires. And feedback inhibition will help regulate the metabolism. Feedback inhibition means that your body reads what's happening and responds. So here we go. Normal binding of a substrate. We have our enzyme with its very particular shape for the substrate. So when this plops in here, so we've got, this is what I call the lock, this is what I call the key. That goes in there, it causes the reaction. If it's competitive inhibitor, something plops in place. So now the substrate can't fit. The non-competitive plants itself in here and changes the shape of the lock. So now the key doesn't fit. Why this is important is due to this. A lot of our drugs, also pesticides and poisons, but I'm going to talk about drugs, as in medicines. A lot of these act as enzyme inhibitors. So these are designed specifically to block certain enzymes from reacting a certain way. Yeah. And there you go. That is chapter 5. Obviously no lab there because we've already done the lab. but I hope that was helpful. It's kind of weird to sit here and do the one that I have only ever taught while throughout class, but hopefully I didn't forget anything and hopefully that worked. So peace out.